Everybody, if you'll take your seats, we'll go ahead and get started. We were trying to let a few folks in registration make it through so they can get in. All right, well, good afternoon. I'm excited to see so many engaged and energetic minds here at UNCW today for today's conference. I expect that together we're going to learn a whole lot of new things about the emerging PFAS contaminants in the Cape Fear region. My name is Stuart Barrett, and I have the honor to serve as the Interim Associate Provost for Research and Innovation here at UNCW. Or as I tell my children, I'm actually the chief geek for the university. Right? That title is just overwhelming. In all seriousness, on behalf of the Chancellor, the Provost, and the whole UNCW community, I want to welcome you to our campus. The University of North Carolina, Wilmington is the state's coastal university where we blend excellent student learning with high-quality research, innovation, and entrepreneurship. In case you haven't noticed, UNCW is a university on the move. Our student enrollment has climbed to nearly 17,000 students, and the Carnegie Institute recently reclassified UNCW as a doctoral university with high research activity, or what's colloquially known as an R2 university. Now, this recategorization is not a fundamental shift in the nature of UNCW. Instead, it's a recognition of who we are and our promise for the future. We're growing the university's research and innovation efforts, and that's exciting for a number of different reasons. First, research and innovation have the power to uncover and build new knowledge, solve complex problems, and transform our world. Second, the growing research work can enhance the quality of the UNCW education for all students. We do high-quality research with students. Let me just give you a couple of examples so that you have an idea of what some of the things we've been doing. Dr. Ibrahimi in computer science has been collaborating with the New Hanover Regional Medical Center to create a virtual reality experience to help prepare children for surgery. Dr. Joseph Long is leading a team of UNCW faculty and students to investigate the response and recovery of both natural and built environments, coastlines, impacted by Hurricane Florence. And Dr. Jennifer McCall and her laboratory are studying the toxins produced by marine algae, designing bioassays to detect the toxins in seafood, and then growing a company, that's Seatox Research Inc., to market and sell these innovations. Now, these are just really the proverbial tips of the iceberg of what's going on at UNCW. And of course, today, we're here and honored to be part of the PFAS Network, which is a collaboration of researchers from, if I counted correctly, 7 universities in North Carolina to study the environmental and health effects of PFAS, or polyfluoroalkyl substances, in the state's waterways. I'm glad I don't have to say that again. Thank you. Now, you're going to hear an awful lot more about the PFAS network and the science and scientific discovery today, so I'm going to leave those details to others. But what I do want to do is point out that this is an example of the advantage of having multiple high-quality research universities in the state. Working together, we're able to combine diverse expertise and experience to consider and hopefully help address multiple issues related to emerging public concerns, such as PFAS. So, in closing, I want to acknowledge and thank the North Carolina Coastal Federation for their leadership and efforts to make today's conference possible. UNCW is excited to help host today's events, and I look forward to learning with you more about the emerging PFAS contaminants in the Cape Fear region. Welcome to UNCW. Thank you. Now I'm going to turn it over to Carrie Allen from Coastal Fed, and she'll get us started. Thank you.
Thank you very much, Dr. Barrett. Good afternoon. My name is Carrie Allen, and I'm the Coastal Advocate for the Southeast region of the North Carolina Coastal Federation. I'd like to thank you for taking the time from your busy schedules and Friday afternoons to be here to learn more about this important issue that affects us all. We have some of the best scientific minds from North Carolina's universities with us this afternoon, and I know I'm looking forward to hearing what they have to say. We would like to thank the PFAS Network for helping to organize this event, UNCW for hosting this forum, and the Orton Foundation for their generous sponsorship. Our sincere gratitude goes to the university scientists who have undertaken these research projects and are sharing their work here today. It was ongoing research led by these scientists that exposed the presence of Gen X and other emerging contaminants in our waters, providing the basis for immediate enforcement action in a state and national focus on this crisis that did not previously exist. The research funded through this study will provide understanding about the nature and extent of the contamination in our waters, the potential effects of this contamination on our human and ecological health, scientific guidance on the efficacy of various treatment options, and much needed expertise in partnering with state agencies to establish a statewide monitoring infrastructure to ensure there is no next Gen X crisis. Forums like this one are intended to provide unbiased, credible scientific information to our coastal community residents and leaders, which is a critical step in informing policy, regulatory decisions, and citizen actions that make a difference in protecting our waters and natural coast. I'd also like to take a moment to thank all of our elected officials who have taken the time to come out and learn about the work of these scientists. Senator Harper Peterson, Representative Deb Butler, Representative Carson Smith, Wilmington City Councilman Paul Lawler, New Hanover County Commissioner Rob Zappel, and Adam Caldwell from Senator Tillis's office are all joining us today. If there's anyone I may have missed, please let me know. I thank our volunteers and interns helping us here today. I'd also like to acknowledge our founder and executive director, Todd Miller, and Lauren Colodi, our deputy director. Founded in 1982, the North Carolina Coastal Federation is a member-supported organization focused on protecting and restoring the coast. We believe every informed opinion matters. We work to keep critical issues, such as this, at the forefront of our collective awareness. Before we get started with our speakers, I'd like to go over a few logistics. As you can see, we have a full program this afternoon. Following each of the research presentations, there will be about 10 minutes of question and answer. To keep things moving smoothly and to maximize our time, we'll be collecting questions in 3 different ways during the talks. First, you can write down your question as well as the speaker's name on the provided note cards. Once written, please hold up your cards and runners will deliver them to the team down here. You can also, if you have a smartphone, go to pollev.com/PFAS, submit your question there, or you can text PFAS to 22333, and then you'll get a registration link to text your question. We realize that not everyone's questions will get answered today, but after the forum, we'll work to get answers to as many questions as we can and post them to our Emerging Contaminants webpage at nccoast.org/GenX. Following the event, we invite you to join us upstairs for a reception where you can enjoy some refreshments, meet and talk with our speakers and other members of the PFAS network, and view some of the research posters. All of our speakers have been generous enough to share their presentations with us, which will be posted to our Emerging Contaminants webpage, along with a recording of today's presentations. Finally, restrooms and water fountains are just outside where you signed in. You can actually access them through this door here. Thank you again for being here, and without further ado, I'd like to introduce Brad Ives to give us an overview of the North Carolina Policy Collaboratory. Quick introduction. Brad is the Associate Vice Chancellor of Campus Enterprises at UNC Chapel Hill and also serves as the university's Chief Sustainability Officer, as well as the Director of the North Carolina Policy Collaboratory. He serves on the board of the UNC Energy Frontier Research Center, which runs the Solar Fuels Initiative, and also a board member of the Clemson University Institute for Parks.
Great, thank you very much, Carrie. It's really a pleasure to be here today, and I know a lot of you, when you probably saw this, said, what is the North Carolina Policy Collaboratory and what does it have to do with water quality? So my goal here today in the next 2 or 3 minutes is to try and give you a good sense of that. The Collaboratory is a really unique institution, and North Carolina's got a long history of doing things that are at the forefront of education, of research, of science, and I think the Collaboratory is yet another example of that. It was a creation of the legislature in 2016, and the idea was to find a way to bring the needs of lawmakers and figuring out how to solve problems in North Carolina together with the research expertise of the entire UNC system. So we were created as a result of the identification of this need. We're housed at UNC Chapel Hill, but we really work across the whole system. And I've got a just a couple of things from our founding documents that I think will help you give you an idea of our focus. So the language says that we were established by the state legislature to utilize and disseminate the environmental research expertise of the University of North Carolina system for practical use by state and local government. And we are to do this by facilitating and funding research related to environmental and economic components of the management of natural resources within the state, and also of new technologies for habitat, environmental, and water quality improvement. And then finally, we're supposed to develop and disseminate best practices and get this out to interested parties. We're supposed to lead and participate in projects across the state. Ultimately, we have the ability to make recommendations back to the state legislature. The term collaboratory, I actually honestly found baffling when our chancellor came to me and said we were going to establish one on campus. And I looked at the two root words and I thought, okay, collaboration and laboratory, this makes some sense. But I went on the internet to look up a definition of it. And the definition I found says that a collaboratory is a center without walls in which researchers can perform their research without regard to physical location, interacting with colleagues, accessing instrumentation, sharing data, and computational resources, and accessing information and digital libraries in a collaborative manner. I think what you see today from the PFAS network will really give you a much better idea of what the collaboratory is and does. What we are doing is bringing together researchers from across North Carolina, from UNC system schools. We've even reached out to Duke University, a private institution, to go get the best and brightest minds to tackle a big problem. And this is a problem that government itself didn't have the capabilities to do. That you'll hear a lot about the use of mass spectrometers. These are very sophisticated instruments that can analyze samples of water, among other things, and identify chemical compounds in those. Universities have lots of these. Governments really don't. And this was a great opportunity for us to come in and take the capabilities that we have within the universities and bring these out to identify a problem. Now, the collaboratory itself, we don't do any of the research. We have a very small staff. As you can tell from my introduction, I do a few other things at the university. It takes up some of my time. We have Jeff Warren here, who's our research director. He's the sole full-time employee of the collaboratory. Steve Wall is our outreach director. He spends approximately half of his time. We also have a number of interns who work with us, and we have one, Corey Cook, here with us today. I hope you'll get a chance to meet all of them later on this afternoon at the reception. But what we do is we go out and we identify problems. Sometimes the problems are identified by the legislature and directed to us, as it was with this PFAS legislation. Then we go out and look for the teams of scientists. And in this case, there was already a group of scientists who had been working together on the problem. The Coastal Federation had done a great job of bringing them together and getting them in front of us so that we knew who the experts were, so that when the legislature passed the legislation to look at this issue, we could very quickly put all of that together. We're also not a regulator. If things come up and you'll hear about issues that can be problematic, our duty is to identify these for government, to work closely with the Department of Environmental Quality, make them aware of abnormal readings. They then will start their enforcement action if they deem that necessary. They'll also be the ones to work on the solutions to these problems. Again, we are just funding research and identifying the problems. So keep that in mind as you hear the presentations today and think about what the role of government's going to be with all of the work that comes out of this. But in summary, that's who the collaboratory is and what we do. In particular here, the state has put $5 million towards this work. The research network is going out and sampling all of the municipal water supplies in North Carolina. This is the first time this has been done anywhere in the U.S., and we hope that this is a really helpful model where we, as we do identify problems, we'll get that back to government so government can figure out what the solution is going to be for everyone. But we do a lot of other things too. We've got projects looking at invasive species like the woolly adelgid, which is killing hemlock trees in western North Carolina. We've worked on the aftermath of the hurricanes, so looking at coastal flooding. We've studied the efficacy of floodplain buyouts. We've done a lot of work on energy storage for the electrical grid. Overall, we funded about 90 projects to this point, and we're always looking for things that are going to be important to North Carolina going forward. So with that, let me thank our host, UNC Wilmington, and to thank the Coastal Federation, not only for this event, but just for being a great partner with the collaboratory. So thank you very much, and I hope you enjoyed today's presentations.
To give us an introduction to the PFAS Network Research Initiative, we have Dr. Detlef Knappe and Dr. Jason Surratt. They need no introduction, but I'm going to give them one anyhow. Dr. Knappe is a professor of civil, construction, and environmental engineering at North Carolina State University. His research focuses on source water protection, identifying contaminants through targeted and non-targeted analyses, and on the development of treatment approaches for the removal of unregulated contaminants. It's important to acknowledge that it was through research by Dr. Knappe and his colleagues which resulted in the identification of a contaminant known as Gen X in the drinking water supply, affecting more than 300,000 North Carolina residents. The 2016 report issued by his research group described high levels of Gen X in the drinking water supply for the city of Wilmington and surrounding counties located along the Cape Fear River. This work was reported in 2017 by the Wilmington Star News, and the rest is history, still unfolding today. Dr. Surratt is a professor and co-director of undergraduate studies in the Department of Environmental Sciences and Engineering at UNC Chapel Hill. His research focuses on atmospheric chemical processes that lead to the formation of organic fine particulate matter. He is also the program director for the PFAS Testing Network project, overseeing all aspects of their research, communications, and support efforts.
All right, good afternoon. It's great to be back in Wilmington. It's been a while that I've stood here in this theater. Last time, I think, was when we reported back on blood data as part of our Gen X exposure study. So what I want to do this afternoon is first introduce the PFAS Testing Network, and then Jason will follow up and introduce the research initiatives. So I wanted to start out by, let's see whether I can work this. There we go. By first introducing a couple definitions, right? So we keep talking about PFAS as if everybody knows what PFAS is. Many of you have heard about this acronym, but it stands for per and polyfluoroalkyl substances. And we want to also answer other questions. Why are North Carolina communities concerned about exposure to PFAS? And then what are we doing as part of the PFAS Testing Network to understand the extent and significance of exposure? So I jumped ahead. So the abbreviation for PFAS is per and polyfluoroalkyl substances. And it stands for a class of contaminants. This class has several thousand members as we know it. The numbers vary. One report talks about almost 5,000 different registered PFAS structures. So right there you can see this is a very challenging problem. You know, we regulate in the context of drinking water treatment 100 chemicals. Here we're talking about a class of thousands of chemicals, and we'll talk about this a little bit later. Very little is known about the toxicity and health impacts of the vast majority of these compounds. Now, why are we dealing with PFAS in the environment, and why do we worry about PFAS in our drinking water? Well, PFAS are being produced in large quantities because they have desirable properties, and there's a big market for them. And in some cases, I would argue we are unwittingly contributing to the production of PFAS because we purchase products, sometimes unknowingly, that contain PFAS, and I'll get into that a little bit in a minute. So some of the desirable properties that we talk about with PFAS are their high thermal stability, so that's why they're very effective in fighting certain types of fires. They're also water repellent, so if you buy some camping gear and that camping gear stays nice and dry, well, guess what? That comes from coating of PFAS on fabric very often. Also, PFAS are grease repellent, so it's a very unique type of chemical. It's both water and grease repellent, and so that is a reason why you often find PFAS on coatings of paper that are used to wrap your food. Food contact paper, why does the paper not get soggy? It's because it's coated with PFAS that are water and grease repellent. And then also the stain repellency is very convenient, right? If you spill your glass of red wine on the carpet and you just get a paper towel and tab it up, well, the reason why you can do that is because there's PFAS coating on the carpet. So two types of these chemicals have been studied in detail. One is PFOA or C8, and I don't like to show many chemical structures, but I thought I'd show you at least a couple. So this carbon-fluorine bonds that you see there, that's what makes these compounds so unique in their character. And PFOA has been used to make things like Teflon and Gore-Tex. So it's a chemical that helps companies make these fluoropolymers that are, in probably everybody's home. You probably have a Teflon pan, you probably have a Gore-Tex shoe or jacket or something somewhere in your house. Also for PFOS, a very similar structure. Both of these have 8 carbons. PFOS for a long time was used as the main ingredient in firefighting foam and also in Scotchgard. The main ingredient of Scotchgard has now changed to a shorter chain PFAS, but it's still a PFAS chemical that's in Scotchgard. So, like I said, you find PFAS in many consumer products, and I always like to point people to this website of the 6 classes that talks about different chemical classes that are undesirable from a human health and environmental health perspective. And so you can learn a lot on this website, but what they're showing here in this pictorial basically is all the different places in your home where you might have PFAS. And when you go and purchase something in a store, there's nothing on the label that says this contains PFAS, right? So as consumers, we're often unaware what we're purchasing, and through these purchases, we're also exposed in our homes to PFAS. It's not just through the water, although the drinking water exposure in cases where PFAS are occurring at high levels in the water are an important contributor to the overall exposure. So, but then I have my but, right? So there are all these desirable properties that make products convenient or user-friendly, but PFAS are also problematic. They're extremely persistent chemicals, and we've somehow known in the environmental community for many decades that making persistent organic pollutants is a bad idea, but somehow they keep popping up. And that's one challenge I think that we need to address in a more broad context. Why do we keep approving chemicals that are persistent? They're also toxic, and many are bioaccumulative, meaning they build up in our bodies, or they're highly mobile in the environment, meaning that they travel very quickly with water, for example. And that also means they're often very difficult to remove in a water treatment plant and get them out of your drinking water. So this bioaccumulation part I want to highlight is one of the reasons why we worry about these chemicals at very low concentrations. They stay in our bodies for a very long time, years in the case of PFOS and PFOA. So if you keep drinking water, these compounds keep building up. So I often hear people say, well, parts per trillion, that's such a low concentration, why do we worry about it? Well, in our bodies, these concentrations are often hundreds to thousands of times higher when we're exposed to these chemicals. So, why are North Carolina communities concerned about PFAS? We're coming up on the 2-year anniversary of the newspaper article that really brought this to the forefront of the community here in Wilmington. On June 7th, Vaughn Haggerty's article came out and basically told people here in Wilmington, you're exposed to Gen X. And these are some of the results that many of you may have seen. We monitored the drinking water and the raw water source over a period of several months and found that PFAS are present at elevated levels and that the drinking water plant, even though it has very advanced treatment processes, is unable to remove these compounds from the water. And besides Gen X, I always like to point out there were other PFAS in the water at much higher concentrations, and that's something that I always feel I need to emphasize. There's been so much emphasis on Gen X because it's easy to say and to remember, but it wasn't necessarily the compound that was dominant in the water or is dominant in the blood of the population here in Wilmington. Why are we also concerned about PFAS? Well, the duration of the exposure has been very long. When this article came out about a week after the initial article saying that Chemours, or before that DuPont, was discharging PFAS for almost 40 years into the river. The encouraging aspect of this story is that PFAS levels have begun to drop quickly after the industry agreed to stop discharging processed wastewater into the river. And so today, PFAS levels in the Wilmington drinking water are much, much lower than what they were 2 years ago. I would say 99% reduction in PFAS concentrations. So that's a success story, and I would credit the actions of local officials and the outpour of the community. You all played an important role in making this happen. Why else do we care about PFAS? There are also air emissions around around the fluorochemical producer, and that has led to groundwater contamination around the plant. And so it's not just a problem for public water systems, but it's also a problem for private well owners. And this came as a very big surprise for the residents that live around the Chemours facility. These wells have been impacted over a radius of at least 5 miles and probably beyond that. And then, it's not only Chemours. There are other PFAS sources in the environment. They include things like firefighting foam, land application of sewage sludges, and also, improper waste disposal. And often that can lead to either runoff from contaminated sites into rivers or leaching of contaminants into the groundwater. And I just want to highlight here at the end that there are numerous counties in the state, and this is really just scratching the surface, where PFAS have been detected in public water supplies. And one that's still very high on the list of concerns is the city of Pittsburgh. In Pittsburgh, PFAS have been detected for over a decade at high levels. In this figure, you can see the shift in the speciation of PFAS. They were longer chain PFAS in 2006, shorter chain now, but the levels are still very high. And guess what? This water flows downhill to Wilmington, right? These PFAS are still in the drinking water source here at much lower levels because of dilution. But this is still an effort that needs to be, or a problem that needs to be dealt with. We have other sources, other inputs into the watershed that are still ongoing. So I'll stop here and turn it over to Jason.
Not sure what's, sorry, just waiting for the slides to switch over. So I'll go ahead and, thank you. Good afternoon, everyone. My name is Jason Surratt. It's a pleasure to meet many of you for the first time today. I just want to say first, it's really been a pleasure for me to be really standing on the shoulders of giants in PFAS research. Many of the people you're going to hear from today are really some of the nation's leaders in this type of research. Since I'm kind of the newer person to this area, I am serving more as a sort of service role in this project as being project manager. But I am an environmental chemist by training, and I'm also a North Carolina native. So I certainly very much care about environmental issues in the state of North Carolina. So my background is in air pollution chemistry, and so I have a lot of experience organizing international field studies looking at the sources of fine particulate matter, especially organic containing fine particulate matter in different cities like Los Angeles and Charlotte as well. So there's different types of smog that my research group looks at across the country and elsewhere. And I apologize, I'm getting over a little bit of a cold here, so I'll try to speak a little bit louder here. So a lot of the great work that's been done by my colleagues here in the state of North Carolina really led to the legislator passing legislation last year, basically outlining that they found that they wanted us to use academic expertise and instrumentation in our public and private universities across the state to address the issue of PFAS, including Gen X, in our drinking water sources. So this is really one of the main initiatives for our project. And so all the universities involved here are shown and also will be represented today in a lot of the research talks that you're going to hear about. So the scope of the water sampling that we were given from the state was that we have to sample every public water supply surface water intake, and there's 190 of those across the state, and we also have to sample those twice. In addition to that, we have one well is selected by each muni for water supply system operating groundwater wells for public water supplies. And so in our state, there's about 688 total wells, and there's 158 municipalities. So together with the groundwater wells and the intake water sources, we have our total sampling universe is 348 sites. In addition to the water sampling, we've also been asked to do additional types of research. One of those includes developing predictive models of private well contamination, also developing or testing existing technologies that can remove potentially PFAS from drinking water. In addition, we are also charged to look at air emissions of PFAS, how they're transported in the atmosphere, and how those PFASs that get emitted into air, how they actually come back to the surface. So that's another big charge that we have. And then the last thing we were asked to do was to prioritize research topics that we felt were needed urgently beyond the ones I've already listed here that we felt needed to be addressed. So these include things like sampling crops, different types of crops across the state to see if they accumulate PFAS, looking at the accumulation of PFASs in our ecosystem, so different types of animals, for example. And we're also doing some health testing using different types of human cells and dosing them with different types of PFASs that we think are important to North Carolina. So that just gives you kind of a sense, and you're going to hear a lot more about those details throughout the afternoon. In the legislation that was passed last year, we are required to do quarterly reporting about our progress. So my office, the Program Management Office, is really leading that effort for the network. And so our first report was submitted in October of 2018. We were able to formulate the network officially late last summer. So basically August 1st-ish, you could think we basically had everybody in place that we wanted to involve across the state. And these quarterly reports are posted to our website, and at the end of the talk I'll have a link there for you that you can write down, and these slides will also be posted, but you can also visit the website to actually download and read those reports if you would like to as well. We are tentatively thinking, well, we were initially told that our final report would be due December 1st, but it's possible now, depending on pending legislation, we might actually get a one-year extension to submit our final report. And in addition to the $5 million you already heard about that we were given by the state, I just want to acknowledge as well as that the North Carolina Policy Collaboratory has provided matching funding, meaning PIs who are submitting external grants to different agencies like the EPA, et cetera, they have been matching those types of funds as well. And so in total, they've added $1.7 million as well. So to kind of give you a sense of how our network is organized, the green is essentially the program management aspects of the project. And so part of the way we decided to have this network managed was we developed a executive advisory committee. And so you see Detlef Kanape, who you just heard from, is one of our co-chairs, and Lee Ferguson at Duke is also another co-chair of this executive advisory committee. And all of these members listed here are some of the leading experts of PFAS in the state. So it's really been great to work with these folks. They really have a good sense of things that we need to prioritize in the network. And so they really help guide myself and the other aspects of program management to make important decisions about what are some of our next steps that we're going to do, and so on. And in blue, what you're seeing is the different research teams that we've assembled across the state. So we have our water sampling team, our private well risk modeling team, our PFAS removal performance testing team, air emissions, and our other applied research opportunities. And so during the afternoon, you're going to hear from one of the members of each of these teams about the specifics of their research projects. And then finally, in the yellowish color here, these are our support teams that we've assembled as well. And so we have people supporting us for risk communications, how we deal with data, and there's lots of data that we're dealing with, and so we're also figuring out ways to share that effectively with the public so that it's easier to digest those. And we also have a synthetic chemist who's involved in making some of these PFAS compounds, because a lot of these you just can't get commercially, so we have to do some of the synthesis ourselves to have access to some of these compounds. So, and for sake of time, I'm going to actually skip the research questions because I know we're kind of running short on time, but you will be hearing all these questions shortly. But what I wanted to quickly get to was just showing you that we already are out in the state doing sampling. So these are real pictures of the different teams that have been doing things so far. So in the top left corner, you can see here our colleagues from NC State and Duke actually doing water sampling in Lillington, North Carolina. We also have some folks from NC State as well who you'll hear from today that have done some work with fish sampling. We also have air samplers across the state, and you can see that we also here at UNCW, we've got a team of folks that are also collecting air samples related to how PFAS might deposit on surfaces, or if they get into cloud water, they eventually rain out, and we can also look at the rainwater. There's also been some alligator sampling. They're kind of near the top of the food chain, and so we've been interested to see how PFAS are maybe bioaccumulating into those types of species. And then we're also doing toxicological assessments. A lot of this work is being led at ECU, and so you'll hear probably some of this today as well. You're not going to hear from these teams, so I just wanted to show pictures of the folks here that are in the audience that you can also speak with that are not going to be a part of the research talks that you'll hear about. So we have many folks from the project management team. We have the risk communications folks here, as well as some of the data management folks. So if you're interested in talking with some of these people, they'll be in the audience and also at the poster sessions. And then lastly, I just wanted to mention that our communications team and our data management team have helped us really assemble this very nice website that's now active and available to the public. So we have the details of the project all throughout these tabs here. So if you want to learn more about the different research teams, the different types of data and tools that we have available for you, as well as different types of resources such as newsletters that we put out every month, sort of just communicating different things that we think are important about what's going on in the network. You're welcome to check this out, and you can also ask to be a part of the newsletter release, so you can sign up for that, and we really welcome anybody who's interested to take part of that. So in addition to this, the communications team has also been helping us really get out our message to the public, so helping us organize events such as this. So we actually gave our first research symposium back at Duke in the fall of 2018. We will have one at UNC in the fall of 2019, updating the public about our results so far. And also next spring at NC State, we'll have another research update about what's been going on. And then also having briefings like this at Wilmington. We'll also do one in Fayetteville and the RTP. But the goal here is we really want to increase the awareness, the public awareness of our study findings, and to create opportunities to have dialogue with all of you about the study and really kind of get feedback about things that we've been doing. And then finally, the other thing that the communications team is doing is holding things like science cafes. And those types of those information will also be posted to our website. But here is Dr. Scott Belcher from NC State doing a science cafe at the North Carolina Museum of Natural Sciences. And so that's another kind of an opportunity for you all to get more information from different types of researchers in our network. And then finally, we've been doing some scientific communications training with our researchers so that they are better equipped to also talk with each of you at a level that you can understand as well about the different results. Some of us scientists sometimes can overdo it sometimes. And then finally, the data management aspects. I just wanted to say our data management team has begun to develop a data hub for the network. And the idea here is this will allow the public to access latest data from the network through different types of visualizations that are interactive. And this will give you data visualizations for the air concentrations of PFAS, the water concentrations, and et cetera. So we're trying to develop user-friendly tools to actually look at those things. And so that once the hub is officially launched, data will be updated as soon as possible from the different research teams. And you can also visit our website under the Data and Tools tab. We will have a site, a link there that you can click on that will allow you to kind of like register for receiving updates. You know, hey, new data was released, and you can be notified of that if you're curious. And if you have any more questions or want more information, you're welcome to email myself or my assistant director, essentially, Wanda Bodnar. We both work together very closely in running the program management aspects of the network, and we're certainly happy to help answer any questions that you might have if you don't get them answered today. Thank you so much.
Thank you, Dr. Surratt, and all of our speakers who have already spoken. I want to just add my thanks. My name is Tracy Scrable, and I'm a coastal scientist with the North Carolina Coastal Federation and the regional manager for this region. So I want to thank you all for being here. We really appreciate it, and I just want to add just a thought about these researchers and this research effort. I have spent most of my professional career working with research scientists at universities, and it is it's been a very much of a pleasure and really impressive to see the level of collaboration within this network and these researchers going back well before the establishment of this effort. There's also a level of personal sacrifice and professional sacrifice, since most of these folks have other responsibilities. And when this all occurred, I see them pretty much, even though they're funded, there are a lot of sacrifices that they're making to make this happen, and a level of personal responsibility and urgency, because they understand that our health is at risk and our community. So I really want to thank them above and beyond that they're funded, that this is very inspirational, and we thank you all. So I'm going to introduce, as each speaker comes up, just what that team's responsibility is, and then the folks who are going to be speaking on behalf of their entire team will give you information on their work. So team number one is the PFAS sampling and analysis team. Dr. Knappe is going to be speaking on behalf of this team. And the overall goal of Team 1 is to analyze all professional drinking water sources in North Carolina for PFASs using suspect screening, I don't know what that is, but he will explain it, non-targeted analysis, and quantitative analysis. Without further ado, Dr. Knappe.
All right, one more time. So I'm happy to talk a little bit about efforts related to Team 1. That team is charged with sampling all public water supplies in the state of North Carolina. So that is a very meaningful effort for me in the sense that we are able to assess whether PFAS occur in the drinking water sources of all the different public water systems in the state. My team colleagues are Lee Ferguson at Duke University and May Sun at UNC Charlotte, and together I'll explain how we together are trying to address this problem. We couldn't do this work without other team members. We have several postdocs and students involved in this, and I want to especially highlight Abby Joyce at Duke University and Noel DeStefano, who's in my group now. And some of the results I'll be showing are from their efforts. So, why are we doing this? So the key motivating question that underlies all this is the question that many people have asked me, that reporters like to ask a lot, is the water safe to drink? Would you drink this water? And this is really a difficult question to answer, and I usually start out with this quote, and some of you who are in my generation or older, you probably remember this quote well. And when I first heard it, I was like, not that impressed by it, but the more I think about it, it actually applies very well to is the water safe to drink? And so, regulated contaminants are the known knowns, right? And that's when you call up the water plant and ask, is my water safe to drink? They will answer yes if all the standards are met, right? And so that's saying for all the known knowns, check, we're meeting the requirements. And so it's that framework that really has guided us for several decades, and it's not a bad framework. I would say drinking water in this country is quite safe relative to many other places around the globe, but we can perhaps do better, right? And so the next thing to think about here is the known unknowns, right? So in the case of Wilmington, Gen X is on that list. It's an unregulated contaminant in terms of having an enforceable drinking water standard. There are many other contaminants that are unregulated that we are aware of, but the water plant doesn't really have a standard by which they can answer the question that you might ask, is the water safe, right? If there is no standard, the water plant really has no measuring guidepost to tell you yes or no. And then, the reason why I think it's so hard to answer this question is, well, if I don't know what's in the water besides all the things that I do know, there may be something bad there and we just didn't know about it. Like Gen X wasn't in the vocabulary of anybody apart from maybe the generation Gen X in 2015, right? Nobody thought of Gen X as a water contaminant. So, this leads to the iceberg analogy that many of you may be familiar with, right? We only see the tip of the iceberg. There's somewhere on the order of 80 to 100,000 chemicals in commerce, but only 200 or so have been tested in a meaningful way for health effects, and only 100 of them are regulated from a drinking water quality perspective. A lot of these chemicals are produced in large quantity, in the order of maybe 7,000 or so. So that number, again, is much larger than the number of regulated compounds. And then even the number of new chemicals that may get added to this iceberg greatly exceeds the number of chemicals for which we have, useful toxicological information. And, we can distill this down to just PFAS. We don't have to just think about the universe of chemicals, and it's still very daunting, right? We may have on the order of 5,000 PFAS, but when a drinking water plant currently submits a sample to a commercial lab and asks the lab, is there PFAS in my water? that method targets 18 PFAS, right? So you get information for 18, but the other 4,900, whatever, are, unknown. They fly below the radar. So that's where we come in, and where untargeted or non-targeted analysis comes in using high-resolution mass spectrometry. And that's still, an emerging area in environmental analytical chemistry. It's not easy. It's not something that you would probably do for routine analysis, but it's very powerful in terms of identifying new PFAS. For example, I'm just showing a structure here. The structure was first identified by Mark Streiner in water, and the publication on this chemical just came out this year. We have been able to measure the same chemical in the blood of the Wilmington residents, and we could only identify this chemical through high-resolution mass spectrometry. And more broadly, so I am going to show a few structures. This is the soup of stuff that was in your drinking water for probably 40 years. All these different fluoroethers. They're ethers because they have these oxygen groups. And I want to give a shout out here to Mark Streiner at the bottom left. It was his work that identified all these structures. Without all of his efforts, we wouldn't have been able to analyze your drinking water in a meaningful way for PFAS. And the other thing I want to highlight is that, in the blood work that we're currently doing with Jane Hoppin at NC State in the lead, two of these compounds have been found in the vast majority of residents in Wilmington. The top is Naphion byproduct 2, we found in 99% of all samples, and PFO4DA in almost as many samples. So all of this is a result of high-resolution mass spectrometry, and that's still an instrument that you wouldn't find in a commercial lab, usually, that you wouldn't find in a state lab that does routine analysis. So the state legislature recognized that there exists a unique capability at academic institutions around the state to identify water contaminants beyond those that are readily identified through routine monitoring programs. And so it basically guided the collaboratory to assemble a team of researchers to address this challenge and measure PFAS in the state's drinking water sources. So the questions that we're dealing with, what are the concentrations of targeted legacy and emerging PFAS in North Carolina public drinking water sources? So we're charged with sampling all water systems around the state. The number is somewhere close to 350. We're still, there are a couple debates over a couple utilities. And we will do repeat sampling. So we're in our first round of sampling, and then we will do a second round of sampling in the third quarter of this year. We also then will look for unknowns, the unknown unknowns. Are there any new PFAS in the water that aren't on anybody's list so far? And so we're going to screen against the list of 5,000 plus structures that are known to us through databases. And then another way to check whether we're capturing the vast majority of PFAS is to do something called a total organic fluorine analysis. So that will tell us how much organic fluorine is in the sample and what fraction of this total fluorine are we quantifying through our targeted and untargeted analysis. So in a graphic, the analysis strategy looks like this. So we have our water sample, and then we run it through a low-resolution mass spec using a targeted list. We have 55 compounds to see, what are the levels of those PFAS in drinking water. Are they there, and if so, at what levels? And then we'll go over here and do the high-resolution mass spec to see, are there other PFAS that are identifiable in the water sample. And finally, the third check is this adsorbable organic fluorine analysis that is a measure of the total organic fluorine. So, you can think of this as an exercise where the goal is to capture this green rectangle. That's the total organic fluorine that may be present in a sample. The targeted analysis may capture just a small piece of that green rectangle, and so that's what our targeted analysis would do. The untargeted may capture that orange area, and then the adsorbable organic fluorine or extractable organic fluorine may capture, depending on which method you use, this or that color, and then this would be the total organic fluorine. So none of the methods will be perfect, but we can get a sense, are we getting close to capturing all the different PFAS that are in the water? So there'll be a quiz on this at the end. So I don't expect you to be able to read all this, but this is the list of compounds that we're targeting right now. So it's a fairly extensive list of PFAS. And then Lee Ferguson at Duke has compiled a database of 5,100 structures against which we will screen our results from the high-resolution mass spec to see, do we get any hits against this database. So the sample acquisition strategy, well, first, North Carolina is a big state, right? So we're, in fact, still hiring a couple people to speed things up. There's a lot of logistics involved, and I'll show you that in a second. But this map shows the surface water systems and the groundwater systems. You see more groundwater systems in blue in the coastal plain, and then more green would be the surface water systems in the Piedmont and the mountains. And we will coordinate sample collection, so Duke will do about half, NC State will do about half, and our goal is to collect on the order of 25 to 30 samples a week. And we collect these samples in polypropylene bottles that are designed for PFAS work. PFAS don't stick to these bottles, and these bottles don't leach PFAS into the water. And we also do a lot of quality control kind of work. It's always very important to make sure that whatever we get it right. So we include trip blanks with our samples, so that means a very clean water sample, laboratory-grade water will travel in the cooler back and forth, and we'll check at the end, is that sample still clean? We also conduct trip spike experiments. We spike a known amount of PFAS into the water. That water sample goes on a trip through North Carolina, comes back, and we check is what we spiked still in the water at the end of the trip. We also replicate 10% of the sample analysis, and then between Duke and NC State, we're actually splitting about 20% of the samples and replicate those to see, are we getting agreement between our labs. So this is a fairly large effort just for this team alone. So there's 3 institutions involved. Duke with Lee Ferguson in the lead, Abby is the postdoc, and then Gordon Gretzinger is a research associate who is a chem informatics person, so he will do a lot of the data crunching. You know, you get incredible amounts of data from high-resolution mass spectrometry, and it takes a lot of expertise and effort to sift through this data and identify potential unknowns. At NC State, that's me, and Noelle would be the postdoc. She's conducting sampling and doing some of the quantitative and non-targeted analysis, and then Zach Hopkins is a PhD student in my group who's also doing the targeted work. And then we have Mei Sun at UNC Charlotte, former postdoc of mine, now an assistant professor there, and her postdoc and PhD student are doing the absorbable organic fluorine and extractable organic fluorine analysis. So this is a little collaboration between our team and Team 7, which is the data analytics team. And so one of the problems we get to solve is what's called the traveling salesman problem, right? How do you most efficiently collect 350 samples? And so, so they ran it through an optimization routine, and they came up with, 27 total trips that last in duration from 1 to 3 days. We collect between 6 and 17 samples per trip, and so far we've collected 58 samples. So this has been extremely helpful. We could have kind of winged it, but this gives us more confidence that we're not wasting our time. So this is one of the advantages of funding a multidisciplinary team, right? I'm an engineer and I know nothing about this traveling sales and optimization problem, really. So anyhow, once we have the sample, so Noelle here is demonstrating sample collection at a tap of a water treatment plant. We then have this sample, we centrifuge it and to separate any particles from the water, and then we analyze it through directly injecting the water into a LCMS, a liquid chromatography mass spectrometry system. And we get these peaks, and the peaks tell us something about the identity of the compound, and the size of the peak will tell us something about how much of that compound is there. What we also will do is, or have been doing, is we concentrate the water sample. So this is a solid phase extraction apparatus. So we pump water through a small amount of sorbent. We capture everything that's in that water sample. Then we rinse it off, that sorbent, with an organic solvent, and we get a much, much smaller volume back. So now it's much more concentrated, so we can see a lot more things. And we run it through the high-resolution mass spec to do the non-targeted analysis. And then here's a pictorial for the absorbable and extractable organic fluorine method. You get the water sample, and you have two options. You do the same thing that we're doing for non-targeted analysis, or you run the water sample through an activated carbon filter, a very small amount of activated carbon. And in the end, then you burn either the carbon or the extract, and during this combustion step, you convert all the organic fluorine to inorganic fluoride, the stuff that a water plant actually adds to help protect your teeth. So, and so that gets done, the fluoride gets analyzed by an ion chromatograph. Right? So then we have all the different pieces, we can fit them together and understand a little bit better how closely are we getting to capturing all the different organic fluorine compounds in the water. So I want to close by highlighting one result that in my opinion demonstrates nicely the value of this activity. So about a month ago, Lee Ferguson, on his way back from Beaufort stopped by the Maysville water treatment plant. And Maysville is a very small town between New Bern and Jacksonville. It has about 1,000 residents. And so, you know, these small systems fly often well below the radar. They didn't have to participate, for example, in the unregulated contaminant monitoring rule. Right? So these systems know much less about the quality of their water than some of the larger systems like Wilmington. And so this water sample was then analyzed both at Duke, and after Duke found, you know, elevated levels of PFHXS and PFOS, we double-checked it in our lab and got the same result. And what we learned was that, you know, they are most likely impacted by some firefighting foam that got into the groundwater. This well is located about a block away from the fire station, so it's a possibility, but we're very sure that it's related somehow to firefighting activity. These PFHXS and PFOS are very much a signature of firefighting foam. So we've been able to engage with the utility and the town engineer to communicate this information, and you know, so they're starting to think about solutions for improving the water quality in this community. So this probably would have never been discovered, or at least not this quickly, if there wouldn't have been the PFAS Testing Network. So with that, I wanted to just thank some other folks. First of all, the Policy Collaboratory for providing the funding to our team. The PFAS management team with Jason and Wanda and Manal and others, they help us, you know, stay on track and help us also with administrative support. Other students in my lab, Julia and Gavin, are both undergrads who are very helpful in terms of just contacting water plants, you know, picking up the phone, getting people to answer the phone, scheduling the trip. So all that takes time. Jake and Lee's lab, and then the postdoc Yuling and Vivek, the PhD student in Mason's lab. And you already saw this picture. This is a sampling in Lillington, North Carolina. So this is also the Cape Fear River, a little bit upstream of you all. So with that, let me stop, and I think we have some time for questions then,
We do. And can everybody hear me? I'm a little mic'd up here, so can you hear me all right up there? Thank you. So just to remind, if you have questions during the presenter's time, if you would, either write them on the card, text them, as Carrie pointed out, or you can send them via email. We will attempt to put 2 or 3, as much as we have time, but please remember also, we will, after this forum, get answers to your questions and post them on the forum website, and you also have an hour and a half after this at the reception to ask questions and get to know these folks. So I have been prioritized 3 questions.
All right.
I feel like a game show host.
Yeah. Can I win $1 million?
Yeah. Didn't you already do that? You deserve it. Have you quantified the level of C8 in the river relative to Gen X and other PFAS?
So in the Cape Fear River, we've been measuring PFOA and PFOS in the water along with Gen X. The PFOA and PFOS levels are seemingly low, on the order of 10 to 30 nanograms per liter down here in Wilmington. But what I find fascinating is that even though these are the levels that we've been seeing been seeing probably since 2013 when we first started collecting samples. The PFOS levels in Wilmington residents' blood is quite elevated relative to the national average. So what this tells us is that even at a level of, let's say, roughly 20 nanograms per liter in the water for PFOS, you can get relatively high levels of PFOS in the blood. So the reason why I'm bringing this up is I question whether the 70 nanograms per liter health advisory level that EPA has for PFOS is sufficiently protective. Even at 20 nanograms per liter, you have blood levels that exceed the national average by quite a bit.
Okay, the next question is, what is the timing to get final results of all of your monitoring? Will it be posted on the PFAS Network website?
Tomorrow? Yeah. No, it takes time. I think, so our plan is as follows. When we see something that is of concern, like the Maysville results that I showed, we will get them out quickly. If we see essentially very low levels or almost nothing, the main thing we will focus on is the untargeted analysis and the screening for the unknown unknowns, and that takes time. And right now I can't quite say how long that is, but we want to make sure that what we put out is defensible, and so it will take some time. The results will get posted on a website, and there will also be a newsletter. Can people subscribe to the newsletter? So that would be one way to keep up with our activities, but otherwise check back frequently on the PFAS Network website.
Okay, just want to, I've been reminded to restate that we will be posting answers to all the questions after the fact that we won't have time to cover today. So the next question, if we can't be sure that the water from a treatment plant is safe, how can we be sure that water pumped out of an aquifer and treated via low pressure RO process reverse osmosis process is safe?
Well, it goes back to the unknown unknowns, and part of it is also the water travels after RO treatment through pipes to your house, and so things can happen there. So, safe is always in the eye of the beholder. Safe doesn't mean zero risk. Safe is something that you're comfortable with, right? And if I drove here today, I could have gotten into a wreck, right? Is driving safe? I took the risk, right? I found it safe enough. So it's really difficult, even with RO-treated water that travels through the pipes to your house, to say, well, is the water at zero risk? That is probably impossible. RO, reverse osmosis, is very effective in removing stuff from water. It removes salt, like sodium chloride, from the water. You can make drinking water out of seawater. So reverse osmosis removes very, very small molecules from water, but it doesn't remove everything. There are certain compounds that can pass through a reverse osmosis membrane. So to really be able to answer that question, you would have to do a very careful analysis on the reverse osmosis treated water, but probably as important, you should sample taps in individual homes or public places to understand what happens as that water travels through the distribution system to the point where you actually take it out and drink it.
That's what we have time for.
Great.
Well, thank you,
All right,
Dr.
thank you.
Canal. we attended the Duke University presentations or conference on PFAS that was referred to earlier, and we were very struck by the fact that the work that our researchers are doing is not being done anywhere else in the United States and worldwide, and so these researchers are also going where they're needed outside of North Carolina on a regular basis, so we're grateful for that work. Okay, Team 2. Team 2 is the Private Well Risk Modeling Group. Dr. Jacqueline McDonald Gibson from UNC Chapel Hill will be presenting for her team. Dr. McDonald Gibson is a professor of environmental sciences and engineering at UNC Chapel Hill with a multidiscipline background in mathematics, engineering, and science to study risk assessment, policy, and communication. Her research focuses on quantifying the burden of disease from environmental pollutants and developing models to predict population health impacts of alternative environmental policy decisions. The focus of Team 2 is to uncover the factors such as PFAS transport mechanisms through the aquifers, influencing the risk of PFAS contamination in water wells near the Chemours plant, and they will be developing a user-friendly, interactive, quantitative model for predicting which private wells are at risk of contamination from the discharge of Gen X at chemical manufacturing facilities. Welcome.
Thank you, Tracy, and thanks everybody for having me here. And before I get started, I wanted to introduce some of the other members of my team who are shown in this picture. Well, actually, they're not all in the picture. Oh, there he is. Dave Genero is a hydrogeologist at NC State who is doing a lot of the field work for this project, and unfortunately, he's out doing field work in Nebraska, I believe, right now, so he can't be here. And then Dr. Javad Rustai, who is a postdoc working with me, who has a PhD in environmental sciences and engineering and also a master's in computer science, and is doing a lot of the computational work for this project. So as Tracy mentioned, the goal of our project is to try to figure out what's going on with private wells around the Chemours site. What is kind of the factors that are driving contamination of these wells. Detlef already showed this map, but this is a map of sampling done up through about a year ago, prepared by DEQ. And here in this map, the red areas show areas where Gen X has been detected in private wells at levels above the provisional health goal set by the state of North Carolina, which is 140 nanograms per liter. And the yellow areas are areas where private wells have been detected with levels that are detectable but less than 140 nanograms per liter. And the green are wells where there's no PFAS detected. And of course, there's Chemours, and these are concentric circles of varying distances. And what you notice is a patchwork, that some wells are contaminated at levels above the health standard and others aren't, and it's a kind of a difficult pattern to discern, to figure out who's at risk. And so what we're trying to do with this project is figure out, what's driving this patchwork. And another question is, how long is it going to take, once releases stop, assuming they stop, for all this stuff to flush out of the groundwater? Now, how do PFAS contaminants get into groundwater from the Chemours facility? This is a conceptual site model, it's called, of an industrial facility over here. And on this picture are the many, many ways that PFAS contaminants can travel from activities that go on at this industrial facility and eventually end up in the groundwater and that people use for their private drinking water wells. And so there's several potential ways. One is to travel through the soil to the groundwater. Over here, here's the factory, and if there's disposal of PFAS contaminants on site, maybe in an unlined disposal area or or one that's lined but leaks, or if there are barrels of things sitting around or spills, these are ways that PFAS from the factory can basically end up traveling through the soil to the groundwater. And I've been working on issues of groundwater contamination for about 30 years, beginning at the National Academy of Sciences in 1990, looking at a class of contaminants then that were emerging concerns in groundwater, and those were chlorinated solvents. And I'll say back in the early 90s, this is really what we thought about. That was the major way that these contaminants that we were looking at and most worried about in groundwater at the time got into groundwater. But there's other potential routes. One is that if there's runoff from the factory that contaminates a river, or if there's discharge from the factory, as there was at Comores to the Cape Fear River, that water from the river can travel into the groundwater, and the travel can go the other way as well. But then what's really new, this is something we really didn't think about when we were worried about chlorinated solvents back in the early 90s, is air as a source of exposure to groundwater contamination. So you've all heard about this already, but the Comores facility, of course, releases PFAS contaminants from its industrial stacks, and they can travel with the wind and then either be deposited or be washed out with rain, land on the soil, and from there percolate into the groundwater. And what's interesting is that it's turning out at other chemical plants in the nation and around the world that air turns out to be an important pathway for travel of these PFAS contaminants from industrial sources into groundwater. This is just a picture from a paper that was published about 10 years ago on what's now a Comores facility, then was DuPont, in Parkersburg, West Virginia. And there was actually there a municipal drinking water well field that was contaminated. These weren't private wells, but municipal wells. And the researchers figured out then that really transport through the air from the stacks of the factory through the air to be deposited either through rain or settling was the major source of contamination. And so our objectives, again, are to try to figure out from those possible ways that PFAS can get into people's private wells, what's really going on around the Comores facility. Why are some of the wells contaminated and some aren't? What are the features of those wells, and the features of the landscape, the weather, the soil? How do all those things come together to influence risk at any particular well? And then one of our other goals is to develop a web-based platform that I'll show you, a little prototype in a few minutes, that private well owners or officials from the health department can use if they want to look in a particular area and a particular well and get a prediction of whether that well also might be at risk. What's the chance that there might be Gen X or other PFAS contaminants in that well? And then the third thing is to figure out how long it's going to take, again, to flush all this stuff out of the groundwater. Now that Comores has curtailed its emissions, there's still this long-term reservoir of PFAS contaminants in the water, and how long is it going to take to get rid of all that? And my part of this project is really parts 1 and 2. Dr. Genero at NC State is doing this part. So at the end, I'll say just a little bit about what he's found so far, and he's done some really exciting work, but mainly I'm going to talk about my part because that's what I know. And so how are we doing this? How are we figuring out, what is really driving this interesting patchwork pattern of some wells at risk and others not at risk? And what we're using is an approach that's pretty new for environmental engineering, which is we are using artificial intelligence. And the National Academy of Sciences in a recent report said that artificial intelligence is like the new electricity, right? We're using it to solve all kinds of problems that we wouldn't have imagined using this for even 10 years ago. One example is medical diagnostics. Artificial intelligence systems now really drive expert systems that help doctors in emergency rooms and clinics try to figure out what is the most likely disease that might be causing their patient's symptoms. So here's just a little schematic of a practitioner in an emergency room who might have access to a computer that's got an artificial intelligence system. And as the patient comes in and starts reporting symptoms, and then the doctor does some tests, you put those results into the system, and out comes a report with an explanation of what might be driving those symptoms, what the underlying illness might be. So in this case, we're looking at what is making our wells sick, right? What are the features that might increase the likelihood that somebody's well is sick? How likely is it that their well is sick? And what does this artificial intelligence do? Well, it takes big spreadsheets of daunting-looking data that might look something like this. This is actually from a spreadsheet of 16,000 rows of data. So imagine this going on for 16,000 rows. And this is actually marketing data for an automobile company. They use artificial intelligence to figure out how can they best position their ads and create the best mix of ads to increase sales. So what we do with these artificial intelligence methods is we try to look for patterns in the data. So this is the equivalent of this giant spreadsheet. Again, imagining this goes down for 16,000 rows. Instead of having to look at that, we have this that looks at how things are connected in these sales data. And what we want to predict here, or this auto company wants to predict, is basically how many sales are occurring. That's right here, and there's all these other factors. Well, where this artificial intelligence stuff really originated was back in the 70s. Who remembers floppy disks? Okay, so there are other people in this room as old as I am who remember that we used to store what we now regard as inconsequentially small amounts of data on floppy disks. And it was really hard to store large data sets. And so computer scientists were looking for clever ways to collapse data so they could store more of it and not need to have such a huge pile of floppy disks. And what they came up with, again, is these kinds of artificial intelligence methods. They realized that if you can identify patterns in data and how things are really related, you need a lot less storage space. And the modern application that we're using is we're looking for patterns in data that relate to private well contamination to see what can we learn from that. And so what are we doing? So Dr. Roustai has developed a wonderful data set with many kinds of factors that we think might be affecting the PFAS occurrence in private wells. Of course, the number one is we have test results from 803 wells in Cumberland, Bladen, and Robeson counties that have already been tested for Gen X, PFAS and PFOA for some of them. All have been tested for Gen X. So that's what we're focusing on right now because that's our largest data set. So that's the first part. Then he's collecting all kinds of information from other publicly available sources, including land cover information. There's some hypotheses that things like tree cover, impervious surfaces, and things can influence that risk of PFAS contaminants depositing from the air onto the ground and getting into the groundwater. Again, looking at soil surveys because the type of soil has a big influence on how fast and how far those PFAS contaminants can travel. He's also looking at other potential sources. So there's debates in some areas of whether is Chemours really the source. So he's also looked for each and every well that's been tested at how many waste sites and landfills